Okay. Let's get rolling in. So, a couple of fungal case studies just to round off our fungal plant pathogens. So we've got a huge diversity, so let's just pick a couple of random ones. In the end, I've picked two obligate biotrophs, but from different, quite distinctly different. So the first one, powdery scab disease of potato. Why? Because I like it, and it's one of our major, in fact it's probably the biggest issue facing Australian potato production currently. It looks pretty ugly too. It's prevalent around the globe, wherever potatoes are grown. It's particularly significant in Tasmania. Why? They've recently developed a diagnostic test that can test the amount of pathogen in soil. So that's been going for about maybe half a dozen years, maybe 10 years now. They've had the capacity to detect how much we've got in the soil. For not completely clear reasons, it looks like Tasmania holds the world record for the amount of pathogen in the soil. We've got levels that are incredibly high compared to most other places, even where the disease is quite prevalent. It's a bit of a perfect storm. I think partly it's because we have a nice mild climate. Lots of other places, the soil freezes, or there's huge amounts of waterlogging and maybe flooding over winter, and so a lot of that can actually eliminate some of the microbes. We've got lovely deep friable soils that is a nice environment, and things tend to survive. So we have very, very high inoculant levels, and it's nasty here. Estimations around about $3.5 million a year just to the processing sector of our industry in Australia. It is unlike, if you remember, and partly this is a good one to contrast with the common scab story we talked about earlier. Common scab was a quality thing. It was a cosmetic disease. This one is obviously cosmetic as well, but it also affects yield. It actually can, and it's hard to get a clear figure on that, but there's at least estimations of 10 to 20% yield loss based on this infection. It's caused by a protozoan pathogen, so from the Plasmodiophora phyla, and it's got a horrible big long name, so it's Spongospora subterranea, subspecies subterranea. Interestingly, it obviously doesn't have sexual reproduction. Genetic variability is actually very, very limited. So there's been a recent study by a group out of Europe that got isolates from all over the world and tested them to see how diverse they were, and they're all pretty much the same. They're all very, very similar, except for the Peruvian region around South America, where there was a reasonable amount of diversity, and that makes sense because that's the home of potato. That's probably the home of the pathogen. That's where it evolved and changed, and then one version got sent around the world with potatoes, and that's kind of the one that's dominated. So good in some ways because we've got limited genetic diversity in the pathogen. It doesn't seem to be evolving terribly much, so control strategies will hopefully be robust globally. Symptoms. It's got three major disease symptoms, of which probably the most important one has been ignored for most of the time anyone's worked on this disease. So the obvious one is the tuber lesions. So this is pretty ugly, and you've got these tuber lesions that will cause cosmetic damage to tubers and reduce its value. You also get these root galls, which are also pretty ugly and nasty in the roots, which may or may not impede root function. They probably do. They're probably competing for resources, but that's another obvious disease thing. What's probably the most important in terms of yield reduction, however, is the zoosporangial infection of the fine roots. So all the root hairs So the infection occurs, you get these fungal structures inside the roots, and that seriously affects the ability of the roots to transport water and nutrients. The interesting thing is these three phases, this leads to this, which also leads to this, but they are independent in terms of varietal susceptibility. So you've got some varieties that are highly susceptible to this phase and don't show any root galling. They've got a little bit of that. There's some varieties that show lots of root galling, don't show much tuber symptoms, they've got a bit of that. Some of them don't show much of this at all, but they might be absolutely nailed in the zoosporangial infection, so they still get yield loss, you just don't notice it because you don't see any scabby spuds. The fact that those are not directly linked in terms of presence of one has, high levels of that has to lead to high levels of the other, has confused people in terms of management by resistance. So they've got resistant varieties that have low tuber infection, but they get nailed with that. Russet Burbank is probably a classic example, again locally, because it doesn't get much tuber lesions. It can, but it doesn't get too much. Not as bad as some. It gets some root galling, not as bad as some, but it certainly gets nailed with zoosporangial root infection, and you've also got a very small, relatively small root system, so the impact on that root is actually really quite strong. Compared to other varieties that might have a much bigger root system and tolerate the disease much better. Anyway, how does it work? The pathogen starts off as a resting structure. So these resting spores, they're called sporosauri. They're actually an agglomeration of lots of resting spores. Actually, I think we've got that in the next cycle. Yeah. So this agglomeration of all the resting spores, so these agglomerated, there's up to several hundred individual cells there that are all resting spores, and inside each one of those is held a zoospore, an infective zoospore. But these guys here are really tough. These guys here can survive in the soil for decades. They can dry them, you can heat them up, they'll survive. They form inside the root galls, they also form inside these tuber lesions. And then at harvest, or when the roots die off or rot off, they're released into the soil. Obviously they fall out of these, when these lesions erupt, they fall out into the soil. They'll also be carried on the tubers themselves. And then they'll survive in that soil for decades and waiting for another crop. So it does negate a lot of the value of rotation for controlling this disease. So that's how it survives. The next phase is how does it move between sites. It moves, obviously, it can contaminate those seed tubers. So once you've got a diseased tuber or contaminated tuber and you move that around, you're going to move the pathogen. So that's how it's really spread from continent to continent, from farm to farm, primarily. But also any other way that moves soil around will also spread it. This is a soil pathogen, so anything that moves contaminated soil will spread the pathogen. Infection. So going back to our diagram here, we've got our sporosauri. They've released what they eventually do when they sense that a host plant is around, they will actually germinate. So they're actually quite clever. So these sporosauri, they're dormant, they sit in the soil for decades, but they can be triggered to germinate based on presence of root exudates. So a potato plant's nearby, it's releasing root exudates, there's certain chemicals that will actually trigger that resting structure to germinate, and it will release one of these guys, a zoospore. So here's our spore, it's got a couple of big long flagella, and that gets released and it swims in the soil moisture, the water in the soil environment, and will move towards and attach to a root. Interestingly, the same chemicals that actually, the same root chemicals that actually stimulate germination also are the same ones that attract it to the root. So this works by what we call a term called chemotaxis. What that means is that these zoospores have chemical receptors, and they can detect these chemicals, and they also detect the concentration of them. So it's been produced from the door over there. There's a large concentration of chemical from that door, and as it diffuses towards me, the concentration decreases. So I detect the chemical, and I go around and I go, oh, it's getting more concentrated. I know which direction to go, and eventually I come across and bang, smack into the root. So it can follow concentration gradients to find the root. Very, very clever again. The reason it needs to do that is because these guys, unlike these guys are very short-lived. There's some evidence to suggest 5 hours. I think it's more likely 2 days. But they swim around for a very short time and then they die. So if they don't find that root pretty damn quickly, you won't get infection. When they do bind to the root, they insist on the root and then they actually eject their cell contents into the root itself, and that's how infection occurs. It likes cool, wet soils. So again, if we're contrasting this to common scab, which looks very similar, which likes hot, dry, this is obviously cool, wet, because it needs to have lots of moisture for these things to swim around in. It's a polycyclic disease. So again, unlike common scab, which you get one round of infection, this guy here, infect the root, it produces these zoosporangia. These zoosporangia produce more zoospores, and they get released, and they reinfect, and they reinfect, and they reinfect. So you get this cycling of infection. So you start with a single infection, you end up with millions of infections throughout that root system. So if you get infected early enough, in a young enough plant, it'll eventually disrupt a very large amount of that root system of the host plant, and that's where you get the major disruption of water movement and yield impact. So the infected roots produce the zoosporangia. These zoosporangia produce more zoospores. They reinfect. Again, cool, wet conditions are required, and that increases infection in an exponential manner. So rather than moving up 1 infection, 2 infection, it goes 1 infection, 2 infection, 4 infections, 8 infections, etc. It moves an exponential rate. So once we've actually got this infection, once we've got these zoosporangial infections in the root, you start then getting impact on the plant itself. So growth and water uptake is greatly affected. Here's an experimental system. Here's nice healthy spuds, nice healthy looking roots, much longer. Here's ones that have been dipped in zoosporangial, in a zoospore solution. You've got some secondary things in there as well, but you've actually got much smaller root systems, much smaller plants as well. They're doing much worse. You will at some point trigger production of galls. This tends to occur later in the infection process. All of that zoosporangial infection occurs first, and then there's a trigger which we presume is related to something like tuberization. When the plant is actually starting to get mature and starting to say, I'm getting ready to go into survival mode myself, that triggers the pathogen to think, I better get into survival mode too. And so it produces these galls which are full of their resting spores, again, for survival. So it forms root galls, and then when the tubers start to form, the young tubers start to form, they get infected by zoospores, and they initially form these blister-like lesions which will eventually burst and release those resting spores, those sporosauri. So you don't get zoosporangia in your tubers, you'll get the infections will form to production of these resting spores, these sporosauri, these things that are actually going to survive in the longer term. And that's the basic life cycle of that pathogen. You'd note at no point did we see any mycelia or hyphae. It doesn't produce any. So the fungal structures, given that it's not really a fungus, are these zoosporangial structures in the root, and the spore structures that you actually the resting spores and the zoospores. Management. So how do we manage it? Again, go back to our disease triangle. The key thing with our disease triangle, obviously we need our susceptible host, virulent pathogen, conducive environment. If we break any of those interactions, we disrupt disease. So what can we do? Not a lot, but anyway, we can do some things. First off, let's start with environment. We've said already that cool, wet conditions are important for disease. So what can we do about that? There's limited, but avoiding excessive irrigation can be important, particularly during that early infection phase, early phase. Avoiding having good soil drainage, avoiding hard pans, that sort of thing can also be important. So well-drained soils that don't accumulate too much soil moisture, and avoiding excessive irrigation. Will that make it go away? No, but it'll stop making it worse. Soil temperature, pretty hard to manipulate soil temperature, but there's a potential at least for altering planting dates that might, planting into warmer soils rather than cooler soils, that may actually reduce that infection, particularly in that early phase. We tend to get excited about the tuber disease, but really it's that early root infection phase that's going to cause you the most yield loss. So not a huge amount we can do about the environment for that particular pathogen, that particular disease. For the pathogen itself, what can we do? We can try and control where the pathogen comes into the system. So one of the major sources is on the seed tubers, on the stuff you plant. So firstly, seed certification is really critical. So make sure you plant the best possible stuff you've got that isn't covered in disease material. It will reduce it, it won't necessarily eliminate it. It's clear there are examples where perfectly clean, healthy looking seed can still be covered with these resting spores, but generally, if it looks clean, it's mostly clean. And certainly planting disease seed will make it, you'll definitely have a problem. Back that up with some seed treatments, can be effective. At the moment, I don't think there's anything registered for control of this particular disease, but there are overseas where there are some chemicals they do apply to seed tubers that will actually reduce the seed-borne infection. So if you've got clean seed, but if you've got any contaminant material on it, you can then treat it with a fungicide and fungicide seed treatment, seed dressing, and that will actually reduce the amount of pathogen further. So that can help stop the stuff coming in from the seed. Trouble is, you've still got an awful lot in the soil itself. So that's only one avenue of the pathogen. The other avenue is what's been sitting in the soil for 10 years and waiting for you to replant your potatoes in that potato crop. So what can you do? Well, one, you can go into brand new fresh soil with your cleanest possible seed. So you're going to a soil that's never had potatoes before. That's a good option. We're running out of those, and most places like Tasmania, most places have grown potatoes before at some stage or other. So finding fresh land is really difficult, and even finding land that hasn't grown potatoes for 10 years is good, but we've already said that this thing can survive that long, so it might still be there. I said they've got these new tools now to detect the pathogen in the soil. So, and people are starting to use that now. So they're actually going into their fields before they plant, and they're testing the field and they're saying, that's got really high levels, that's got lower levels. Between those two paddocks I might plant, particularly if you're a seed grower, you want really clean stuff. I'm going to go for the low level one where it's going to have less pressure. So for instance, some of the processing companies have particularly done that. When they're planting their seed crops, they'll test all of the paddocks, and they won't plant in a paddock that's got high levels. They just refuse to. And they have reduced the amount of this pathogen in their seed quite dramatically, just by that one strategy of picking fields that have lower levels to start with. Chemical treatments, again, nothing here in Australia, but overseas there are some chemicals that they do use that have limited efficacy but can work. The issue being, once again, that that resting structure is very, very tough. It's actually very tough and almost impervious to a lot of the fungicides too. So with the fungicide treatments, they tend to be killing the zoospores, not the resting spores. So that's fine. You know, you put in a pre-plant fungicide, for instance. You've got your potato. Those that are germinating, they swim out and they get killed by a fungicide. But the fungicide doesn't last forever, and eventually it will run out or won't fully saturate the environment, and you will get zoospores releasing from these dormant sporosauri that weren't killed that will start the infection. So it can be useful, but it's certainly no silver bullet. There's no chemicals that actually work brilliantly. Rotation, look, it's still better than nothing. The longer rotation the better, because naturally they survive for 10 years, but there is some microbial decay over time. I think there was some modelling to suggest that, you know, maybe 15 years, 20 years, it should in theory crash down to below detectable levels, but I suspect there's other factors that will intervene in that process. And the other factor is controlling your weed potatoes. So after you harvest potatoes, you often leave behind a whole bunch of little ones, and then they grow and they actually keep propagating the pathogen in the intervening years. So you don't actually have a real rotation if you don't get rid of all of your volunteer potatoes. But where can we really have a go at this? If we go back and think about, this guy's very, very tough and pretty hard to do anything about. This guy here is really, really weak. Lives for maybe 5 hours or 2 days, and is certainly susceptible to everything we throw at it, and needs wet conditions. And then once the encystment starts, once actually the infection goes, then it sort of starts that process, and the polycyclic process starts. It's pretty hard to control. But if we can then reduce that interaction between the zoospore and the host, that can also be important. So what can we do? Well, actually, it's one thing we've been starting to play with. We haven't taken it to, well, we're only taking it to the field now too, is trying to understand that whole process of germination. So you've got those resting structures, and they're sitting in the soil, and they're clever things. They sit in their soil and they're going, well, I'm just going to wait. I'm going to wait till a plant comes along, and then when the plant comes along and releases these chemicals, it tells me to wake up. Then I'll wake up, and then I'll infect it, and then that's great. What if we trick it? What if we actually hit the soil with a chemical stimulant, a root exudate, in absence of the plant? So you wake up the zoospore, or you wake up the sporosauri, the zoospores swim around and go, yay, I've got 5 hours to find a plant. There isn't one. And then they die. So we've actually been looking at that process, and there's a range of, you can't see it here, but there's a range of amino acids and other things that actually stimulate zoospore germination. And at least in artificial systems, that kind of does work. So here we go, here's our pre-treatment, here's our post-treatment. So to start with, if you just put distilled water in, obviously there's no real difference. If you put one of these stimulants in, after about 6 or 8 weeks, we've crashed the amount of pathogen down in that soil system by just waking it up and let it swim around and die. And so that's actually now trialling that on at least on a field basis to see whether it's going to be remotely useful. But taking beyond that, part of that is also thinking about how do we manage it. That was over about a 6-week period. Where I think that'll actually will seriously work, it's all about timing. What we tend to do when we're trying to manage this disease is we're about to plant potatoes and we go, what do we do to stop this disease? We've got inoculum in the soil. How do we stop that waking up and killing our plant? What treatments can we apply a couple of months before potatoes to make it healthy? It's too late. way is, I've just planted potatoes, I just got nailed with powdery scab, that's horrible. Oh, I don't want to think about potatoes again, I'm about to grow carrots. Well, that's when you start managing your powdery scab. So if you actually go in and use these stimulants or use other processes after your potatoes, where you've got very high levels, and then crash them over a number of years, so by the time your rotation comes around to potatoes again, instead of having high levels of these things sitting in the soil, you actually have managed to crash these down to levels where you're not going to get disease. And so timing, I think, is actually going to be the critical factor for controlling within the rotation. And there's good data already from some of this work where people are measuring pathogen content over the soils across rotation to suggest that that's actually feasible. The other thing which we haven't started yet, but we're kind of keen to do is, again, look at these signals. We talked about the chemotaxis, where the pathogen can sniff out the chemical and work out where the root is. So what we're actually about to start to play with is, can we do a bit like the pheromone traps in orchards? Can we actually confuse the hell out of these zoospores by putting in artificial roots, by putting in compounds? This is actually an absorbent clay particle that's filled full of one of these attractant materials, and here's all these zoospores that are swimming towards it. So if we can actually create a whole bunch of these compounds and actually put them through the soil profile, and these spores wake up and then they don't know where the root is because they're swinging and hitting these things instead of the root, can we actually use that to reduce the amount of infections that occur? So that's I'll let you know if that works. It's going to be cool. The other thing from a host perspective is resistance. Now there's nothing that's immune, and we know already that resistance to root and tuber is there is some there, but nothing's immune, but it's also not linked. So that if a lot of the literature in terms of resistance to powdery scab is all based on the tuber observations, because that was the easiest thing for the breeders to observe. Unfortunately, what we've got now is we've got these resistant varieties that actually are quite susceptible to root infection because they didn't realise they weren't linked. So now there's a lot more effort going on to, can we measure root infection too, because that's actually more important in terms of yield. So there is some variation, but nothing that's perfect. One thing again that we're actually just starting to look at, which we hope will be quite useful, is that where those do vary in their resistance to root infection, it seems to be at this point where encystment binding occurs. So firstly, if we start looking at that whole encystment process, we can get a rapid test tube assessment for resistance. So instead of having to plant a field trial to assess resistance, which is really difficult, we can actually just take a little piece of root, dip it in with some zoospores and work out whether they attach or not. But also the key thing is then to start to look at that whole binding site. What are the proteins that it binds to, and can we actually use that to breed for varieties that don't have those binding proteins or have less of them, or even block them? Any questions about powdery scab? Okay. Round two. So let's go above ground and let's go to a cereal instead. So case study number 2 is a rust of a cereal. So black stem rust of wheat. Again, a really, really important disease. So probably the most important disease of wheat and barley, arguably, globally. And obviously wheat and barley is, wheat is yeah, it probably is. I think wheat is probably still the #1 staple around the world. Rice, I don't know, which one comes first, wheat or rice? Probably rice second, I suspect, I'm not sure. Potatoes come in as number 3 from a human perspective, after wheat and rice, 4 after maize if you include animal feed. Anyway, it's by and by. So wheat and barley together is about 25% of food production. 5 billion US a year lost to the various rusts that affect the cereals. That's a hell of a lot of money, but obviously there's a hell of a lot of cereals grown globally. This particular pathogen is caused by a Puccinia graminis, forma speciale tritici. It's a basidium mycete, so one of these ones that has those clamp connections. It's a rust fungus. It's an obligate biotroph, so it has to live on a living plant. It can't grow on dead material. It can't multiply on dead material. It needs to live and associate with a living host. So it's one of these ones that forms these haustoria and sucks out nutrients from the cells. It is macrocyclic. It actually has 5 different spore types. I think we had that slide earlier on which detailed those 5 different spores. But here they are again. You've got these pycnidiospores, esiospores, uridiospores, teliospores, basidiospores. And they vary in whether they are sexual or asexual. So whether they are a single nuclei or whether they've actually fused. So these are the sexual spores. They've actually fused to be double haploid.
What's the difference between an N plus N and the 2N?
So that's where you've actually got that they're still single N, but they actually haven't fused yet. So it's actually producing the different structures that which you will, the male and the female versions. So symptoms: stem rusts attack all the above ground plants, obviously the stems and the leaves, but also the flowers too. They produce less tillers, fewer seed, and can kill plants, but that's not that common necessarily. Clearly it affects photosynthetic capacity, water and nutrient use, leads to major yield loss. The most obvious symptom is there's orange pustules, orange or red pustules on the leaves and stems in the spring and the summer. In winter, these look black. It's a different spore type. So this is reflecting your different spores at different times. So it's called black stem rust because you've got these black spores in winter and autumn versus the orange ones in spring and summer. The disease is heteroecious, which means it needs more than one host. It doesn't have to use both hosts. It can actually get a pathogenicity cycle on a single host, but it won't go through its entire life cycle. It won't go through production of all the different spore types unless it has these 2 distinct hosts. So this pathogen infects cereals, but it also needs barberry, which is a completely different plant, obviously, as its alternate host to complete its life cycle. So its primary host is wheat and barley, alternate host is a barberry plant. So how does it work if you've got all of those present? As I said, if the barberry is not around, you can still get cyclic infections in wheat, but you won't get the full gamut, or you won't get all 5 spore types produced. So the life cycle. In spring, in the barberry host, you'll have these basidiospores. They land on the barberry, they infect. They form pycnidia structures on the leaves, and these release the pycnidiospores, which will be released, and also these receptive hyphae, which are also infection structures. And as I said here, it can be N plus N means you've got the different mating types. So as I was saying before, so if you've got mating type 1 or you've got multiple mating types. The hyphae, which can have the different mating types within it, grows through the leaf, in spring, produces an acium, and that can produce an asiospore, so the second spore type. So we've got our pycnidia on the top here, the pycnidiospores, and we've got a different spore type on the other side of the leaf, the lower side. These guys, so the pycnidia go off and reinfect barberry. These guys can no longer infect barberry as a host. That's where they have to start moving into their cereal hosts. And they're wind dispersed. So the ichthyospores blow off and land on a wheat plant, they germinate, and they produce an iridium, which looks like this, where you've got the leaf bursting open and you've got these iridospores being produced, round and oval, each on a single stalk. And this is known as the repeating phase, because this is where you can get cyclic infections of wheat plants. And then, obviously they're also the orange rusty structures you see. And then towards autumn and winter in wheat, you'll get production of a sexual structure, this teleum, which is full of teleospores. That's where they undergo sexual recombination. They now 2N instead of N plus N. And these guys are the black spores. So these are the survival structures that can overwinter between crops. Teleospores in spring, they're usually still on the wheat plant. They'll germinate, undergo meiosis to perform a basidia and the basidiospore. And they land on a barberry plant, which complete the life cycle. So horribly complex, multiple spore types, multiple hosts for a disease, particular disease. So you can, how has it evolved? It's sort of evolved to survive between presence of cereals and a perennial, got a perennial alternate host, so it can part of its life cycle in the barberry host, which is perennial, and then it can move into the annual cereals and spread quite rapidly through that system. So it's quite interesting and complex versus some of those much more basic diseases or pathogens like our bacteria that really just kind of invaded, produced a toxin, killed stuff. So that is the life cycle diagrammatically. So again, you start with your barberry host, produces these pycnidiospores and receptive hyphae. They lead to infection, produce these aciospores in the barberry still, and then they get into wheat at that point. Infect, produce these iridospores. You get your sexual recombination, these sexual spores as teleos, and then produce basidiospores, which then go back into the barberry. So, what do we do about it? Or what do we know about its life cycle or the factors that it prefers? So, stem rust likes warm, hot, mild conditions and wet leaves from rain and dew. So we're talking 25 to 30 degrees, fairly warmish, which is not uncommon in wheat growing regions, and mild nights around 15 to 20, so it doesn't like very cold temperatures, but it does like dew or wet leaves for that spores to germinate and to penetrate. Remember, we were talking before about the need for spores to absorb moisture, to swell, to germinate, to infect. So this is much the same. So it's ichthyospores and the iridospores all need free water for germination, as do other spore stages. Infection primarily occurs through natural openings, through the stomata, but once they get in, then they actually form those hostoria to invade and interact with the host plant and sequester nutrients from within the cell of the infected plant. They can move around very, very easily. These spores are highly mobile. These iridospores, particularly, this is the orange replicated form, the one that actually allows infection of wheat from wheat. These guys can get into the atmosphere and dispersed over very large distances. So, obviously within a field, most will be plant to plant to plant within there, but it can actually get blown up into the wind currents and move huge distances. The biggest distance recorded so far is around about just under 700 kilometres, which would have been the equivalent of moving from South Africa to Australia. So it's, it can go quite a bit. And again, in the US system where they monitor it, you'll see these waves of epidemics moving as it moves up the coast. So it'll go from the south to northern US in about 50 k's a day sort of movement. We mentioned before, to do the whole life cycle you need both hosts, but you still can get disease in absence of the barberry. Instead of going through that whole the suite of spore systems in the barberry host, you actually have the rhedospores surviving in relatively warm conditions, and then they reinfect the cereals in the next year. So again, that's a system that happens quite commonly in Australia. Management. It's really all about host resistance. So we're talking about broad acre. One can't afford to use fungicides terribly, over that level, and they're not necessarily all that effective. It's all about managing the host pathogen interaction and getting resistance. So from the 1950s, and this guy Norman Berlag is one of the pioneers of wheat breeding. They started breeding for resistance to stem rust, and there's a number of resistance genes that have been discovered, over 50 odd to date. But of course, as soon as you put up develop a resistance gene, you create a challenge for the pathogen to overcome. And these pathogens, because they've got sexual phases too, have the capacity to overcome that and develop resistance breaking strains that can overcome the host resistance. And generally they do so. So by the time a breeder's brought out a new resistance, it lasts 1 or 2 seasons before resistance breaking strains occur and then start to break that down. There are some resistance genes that are more robust than others and can hang around a bit more, and increasingly what they're realizing now is even some of the ones that have been defeated, some of the genes that are no longer, that strains have been developed against, resistance breaking strains have been developed against, you can reuse them if you stack them with other genes. So if we get rather than a single gene resistance, we get multi-gene resistance. And it's, I guess it's analogous to, here we've got our host plant, here we've got our pathogen, and we're trying to stop that, so resistance is a dirty big brick wall. Bang. And the pathogen kind of beats away at it until it eventually knocks its way through, and once it's through the brick wall, there's nothing to stop it completely utilising that host. If we've put a couple of brick walls, it's got to get through 2 barriers. If they've got different resistance mechanisms, if we put 3, if we put 4, it just makes it more robust. Even if those are defeated genes, so like, okay, yeah, you can kind of jump over that hurdle, jump over that hurdle, jump over that hurdle. So jumping over all 4 hurdles is just too hard for it. It can easily defeat that one, can defeat that one, but the strains that have defeated those 2 are not robust enough to defeat a 3rd. And so they're discovering that by stacking resistance genes, that can actually help make them a bit more robust as well. But, so management of stem rust, as I said, is primarily through breeding, and breeders, full-time breeders going crazy at trying to get more and more resistances all the time. But there are some that are actually still quite relatively robust. Until, particularly this gene, the SR31, which is really quite valuable. until 1999 in Uganda, there was a new strain appeared, the UG99 pathotype. And it can defeat that particular gene, which was most dominant in Africa at the time. But it's also really, really, really nasty. So a particularly severe disease that actually completely killed yield. So grain, the heads didn't fill, there was no yield. It was actually a very, very aggressive strain. And they're going, well, okay, Africa is, wheat's important, but not nearly as important as cassava and maize and other major staples. But there are other parts of the world where wheat is incredibly important. And looking at that, the way things could move, it's not terribly hard to imagine it getting into particularly Northern Europe and Russia, where huge amounts of wheat are actually grown. And you can actually see, these are dates here where movement of some of these strains. So that was actually a different strain, how it moved in the past and got its way up and around and into China. If we look at the UG99, so this is where it's obviously where it started, and it's actually started to spread and even down into South Africa too. But it has actually appeared in the Middle East as well, and so it is moving. It's moving by air currents. It's also potentially moving by movement of materials. And now the real concern is it's going to get into Europe and into the really major wheat production regions of the world. Is it changing? It is evolving as it's going, but the major strain is still dominant. If you look at the red colour, so the red colour that originated here is obviously still the strain up there, but some of the strains that have gone further south have evolved. They're still nasty and they're still resistance breaking, but it is evolving too, because these things don't stay still. So what can we do about it? In fact, some of the cereal breeders have said it was their oh shit time. They're going, everything we've got, this defeats. We don't have any resistance genes that work. And so it's become really quite problematic. So initially, they whacked huge amounts of fungicides around to try and control it. Bill Gates busy tossed a whole bunch of money at it. They have eventually managed to get some resistance against it. And this is starting to be used now. It's not necessarily the world's greatest variety, but at least it's there as a means to actually try and combat the spread of this disease. In Australia, most of our varieties are susceptible. So if it did arrive here, it would be huge. And as the new variants are occurring, even some of the ones that are less susceptible are probably still more still likely to be susceptible if it does arrive. So will it get here? It probably will eventually, because they're almost impossible to keep out, but they can travel huge distances in the air. So there is a real risk, and there are monitoring sites in a lot of the cereal funding agencies actually have these sentinel sites around Australia where they can actually monitor them regularly to actually see what new things potentially are blowing into Australia. And these are really critical as an early warning system if things like this arrive. Okay. Any questions?